Hey, good morning and good afternoon to those joining us today from other time zones for our webinar on NAM-based prediction of respiratory toxicity using human and rat airway cultures. This webinar is being recorded, and all attendees will receive a copy of the recording along with a poster authored by MatTek and Charles River that was presented at this year's Society of Toxicology meeting. Our presentation today will discuss an ongoing project between MatTek and Charles River to develop and implement a new approach methodology inhalation toxicity test to reduce reliance on traditional animal research methods. Our presenters today are Dr. Mary McElroy, the head of Discovery Toxicology and Pharmacology at Charles River Laboratories, and Dr. Sayum Ayuni, Chief Scientific Officer at MatTek Life Sciences. Please join us for a Q&A with our presenters after the presentation, and feel free to place questions into the Q&A section of the chat throughout the presentation. With that, I'll hand it over to Dr. McElroy to begin.
Thank you very much for your nice introduction. Can I check that you can hear me okay? So
Yes.
Yes, fantastic, good. Thank you very much for the nice introduction. I'm delighted to be here with Sayum talking about our progress on these NAM-based prediction models of respiratory toxicity. Next slide, please. So just as to give the running order for today is that I'm starting off with a very general introduction, including why CRL is working in this space, and also about introducing the origin of our American Chemical Council Grant Award. And then Sayum will take over to talk about an introduction to NAMs and how the grant study design protocols were developed. I'll return to talk about the results, and then Sayum and myself will be available for discussion and questions. Next. So I'm sure most of you have probably heard about Charles River. We are a large global company and we're located across the world. We've done many IND applications for approved drugs, but we are much more than an animal development company and a pharmaceutical safety testing company. We also work in the chemical safety assessment space and in manufacture and in discovery. And this leads me to this a specialized group or initiative called AMAP, or Alternative Methods Advancement Project. So AMAP is Charles River's initiative dedicated to pursuing scientific and technological innovation that helps fulfill our commitment to the 3 Rs and the development of alternatives to animal testing. And why now? Well, I think Charles River and the society in general sees the great advances in science and technology that have brought industry to an inflection point. So moving away from ideas associated with toxicology from the 1950s and looking forward to what we can do in terms of in the context of rapidly developing cell biology and developments in AI. Next slide, please. So to give you an example of what the AMAP group covers, in terms of the 3 Rs, we are looking at the inclusion of virtual control groups, using organs on a chip to better detect in vitro genetic toxicity. And also within the discovery space, we are looking at human gut and immune organoids for both safety and efficacy testing. Next slide, please. And within this sort of use of complex in vitro models, where this project sits in collaboration with MatTek, over a year ago, we applied for a joint grant from the American Chemical Council, and we were very privileged to be awarded that grant to advance respiratory tract cell cultures for risk-based decision-making. And this involved also a collaboration with Battelle. And next slide. So just since we're talking about NAM, so there is a recognition that the NAM definition can mean different things depending on publication or approach. So we are using the new method. We're using the ICVAM definition, new approach methodologies. That refers to any technology, methodology, or approach or combination thereof that can be used to provide chemical hazard or risk assessment that supports the 3 Rs. Next slide, please. So I mentioned the American Chemical Council, or ACC for short, and they have a long-range initiative, an LRI grant, and this was awarded to ourselves and MatTek. So who are the ACC? Well, they are a large stakeholder group for more than 190 companies engaged in the business of chemistry. They're an innovative economic growth engine that is helping to solve the biggest challenges facing our country and world. And the objective of this grant was, the purpose of this long-range initiative is to advance the development of an integrated approach to the testing and assessment of chemicals and to evaluate the potential to cause portal of entry toxicity in the respiratory tract. Now, this initiative will have come about for many reasons, not least because of the ethical considerations of using 3 Rs, but also the rapid developments in the development of human in vitro models that resemble the human structure, particularly of the upper airway. So companies like MatTek and Epithelex that have put time and energy and years of development to a point where they can deliver a reproducible product for use in safety assessment. And also because the regulatory authorities are, in all the guidelines, say the best science. And in some cases, these models are being shown to generate human relevant data for human relevant safety assessment. Next slide, please. So our grant is a collaborative grant, and I think all the people, the major scientists within this grant come from different aspects and different expertise. And I want to just point out some of the key participants in the grant. And the first, I'd like to mention Joe Wallace, who's in my team. Joe, who some of you may know, has worked in the space of complex lung 3D models for over 10 years, both for across chemicals, cosmetics, and even drug safety assessment. These studies are run for discovery. They're also run in the GLP space. Myself, I have a long, many decades background in all things lung related, largely in vivo, but with a focus on lung alveolar epithelial biology. But we are within Charles River. We work closely with the pathologists who are in vivo pathologists who are making kind of new algorithms to help us quantify pathological cell damage in our 3D models. And also critically for this grant, we're working with the in vivo inhalation technologists and scientists to take that in vivo learning to ensure robust in vitro delivery of test aerosols. From the MatTek perspective, MatTeks are experts in tissue models development, lung and many other tissues, pioneers in fact, and have been involved in many studies validating and developing in vitro models for safety assessment. And key to this grant also, I'd like to mention Dr. Richard Corley, or Rick Corley, and Kevin Battelle, who are involved in the modeling, if you like, from in vitro to in vivo. So unlike safety assessment in other tissues where, let's say, you put 10 micrograms on the surface of skin or eye, the lung is different in that with a test aerosol or inhaled aerosol has lots of different particle sizes, and the size of these particles or the aerodynamic diameter depends where the particle or the compound will land within the lung. So large particles, say 5 to 20 microns, will be the upper airways, and smaller particles will be more delivered to the lower airways or the alveolar region. So when we're thinking about risk assessment and we're thinking about vitro to in vivo correlation, we have to model the possible aerodynamic particle size of the aerosol at different ranges. And for that, we need computational modeling and expertise by Richard Corley. Next slide, please. Thank you. So this is a summary of some of our key objectives that we put together on the grant. We'll be talking about results from the first objective today. So the first objective really was to evaluate a panel of test chemicals, 10 in fact, to evaluate the reproducibility and predictability of a single application of chemicals in 2D and 3D models, rat and human. And we were looking for what we considered point of contact toxicity chemicals, i.e. those chemicals that would be directly damaging to the airway epithelium, the upper airway epithelium, without, in this scenario, the necessity or the involvement of immune cells. That would be, I think that would have been a very important point too, but we thought was out of scope for what we were applying for. So in this first objective, then we would compare 2D versus 3D sensitivity, and we would compare rat versus human. So the other objectives, which we'll speak about later, would include repeat application, or aerosol delivery, the dosimetry models I have mentioned, multiple donors, and eventually develop a prediction model. And this nice diagram on the right shows that we are looking at in vitro animal to in vitro human, and then we'll be in the future later on as we develop through the program, we'll be able to compare our in vitro assessments with in vivo data, because the chemicals were chosen to have the best quality animal data available, so we can provide confidence in the model in our predictions, hopefully. Next, thank you. So this is the final slide before Sayum talks, we are rooted in making this towards a regulatory acceptance, or to help towards an OECD guideline. So there are various different regulatory authorities have proposed frameworks to accelerate the development. And this one is from ICVAM. And the key idea here is the context of use, that we are developing a model to answer a very specific question. And in this case, it's point of contact toxicity for inhaled aerosols, probably in the larger upper micron range, so they will land in the upper airways rather than the lower alveoli. We will be using a relevant biological model so it's an upper airway model, and so and we'll be looking for, in the first instance, of functional readouts. So in the upper airway, one of the key factors or key properties of any epithelial barrier is intactness. It is a barrier, so the barrier properties, that's a key biologically relevant readout. We'll be running to GLP standards, so protocols and deviations, and SOP-driven and combined protocols. We'll work with collaborators, and we'll publish, and this webinar is part of this process. And the grant will approach the technical characterization, the reproducibility and predictivity. Next. Thank you. And that I'll now pass you on to Sayum.
Okay, can you hear me?
Yes, Sayum. Perfect.
Thank you, Mary. So MatTek is a tissue engineering company, and we have been producing about 17 different types of tissue models, and we have been shipping them out to customers and researchers globally. And we have a global presence. We deliver tissues to North America from the Ashland facility in Massachusetts, and then we have a center in the production center in Slovakia to supply for European Union countries, and also to some extent to extend their hands to Asian countries. We have now extended our presence also in other countries like China. So we have started opening a new facility there, to produce some of our tissue models for the customers over there. We have distributors in Europe, in Asia, like Japan. Corabo is one of our distributor. So we have our availability worldwide is great, and we have expanded our tissue models to be distributed everywhere. Next slide, please. So why do we make this in vitro tissue models? One of the reasons is that a lot of drugs has been tested since the 1938, where FDA, since the inception of FDA, the drug testing requires, it has to pass through animal testing. But animal models display limited translational values, and a lot of drugs are failing. So about 90% of the drugs that pass the animal tests fail in the human clinical trials. So there is a lack of translationality in the models. So continuing failures in drugs and withdrawals of compounds even after they hit the market is so expensive for the pharma company. And as a result, we need transitional models that can predict human responses in the right way, and that can reduce time and cost and also human suffering. And there is also species differences between humans and rats. Most of the data is coming from rats. And animals, rats and humans are genetically different. We have differences in our molecular structures, immunological functions, and also cellular responses. So we intend to show that most probably by using the right model to predict these drugs, then we can increase the efficacy and safety of the drugs, and also we can reduce time and cost for drug development processes, which is expensive currently. If you want to make one drug, it takes about 12 years, and also it requires a spending of about $2 billion. So alternative methods are critical. Next slide, please. So, but we have, the problem is not coming up with new ideas and new concepts. And the difficulty lies in much on the changing of human mind. A lot of people since the 1938, all of the pharma companies have been collecting data on their pharmaceutical compounds using animal models. That was the FDA criteria, that they have to use animal models before they take it into the humans. So even though there are a huge 90% of the drugs are failing, it's hard to change human minds, and also to convince the regulatory bodies to adopt new techniques and new technologies as they come in. So this is an example. Now, for example, the skin model has been developed by MatTek in 1993, and then the pre-validation study for regulatory bodies was started in 1999. And then from 1999, keep that date in mind. So from 1999, then we have to go through protocol optimization phase that took about 3 years between 2001 and 2004. And then the full validation studies takes place from 2004 to 2007. And there is a protocol optimization period of another 2 years. And the adoption in Europe by the Organization of Economic Cooperation and Development in Europe, globally, it's accepted as a standalone method for the skin irritation testing in 2009. That takes a lot of, a total of 10 years to get a regulatory approval. That is in Europe. Europeans was the first one to adopt it. But it is now 2025, and the FDA has never touched it still. So there is no approved method in the United States, but in Europe, it's accepted method. So we have a big challenge, a big to cross. The next slide, please. So this is how the history looks like. For example, European Union adopted, finalized a ban on the sale of cosmetic tests on animals, thereby creating a significant need for alternative methods in 2013. But the regulators in the United States never touched it again until now. So numerous countries have adopted it in Europe, and Europe is the lead in this adopting this NAMs or new approach methodologies. In 2020, FDA, there was a guideline that uses animal test, which is the mouse assay. The FDA released a guideline that the long accepted local lymph node assay for skin sensitization testing of topical products was no longer necessary, and provided the recommendation that alternative cell-based methods with adequate data can be used. But with that, there is no formal acceptance of methods, but here and there on a case-by-case basis, so a few things have been happening. But that is at least a little bit going through the process. And in 2022, there was a FDA Modernization Act, which was passed by the Congress and Senate of the United States and signed by the President. But it was a guideline. Again, it has never been used, picked up from there. So again, now we are 2025, and still we are now coming up with the FDA Modernization Act 3.0, and this new bill requires the FDA to establish a process supporting the use of NAMs to reduce and replace animals for non-clinical research. But with this type of thing, but most of the generations of drugs now coming into the market that are being tested are like cell therapy, immunotherapy, are human specific, and the animal tests are invalid for this type of tests. So there is a great need for this type of the in vitro tests to be adopted for regulatory purpose. But there is hope that the US EPA there is a 30% chance that the new approach methodology can be adopted by 2026. And European Union with the European Chemical Agency, they have a partnership for the assessment of risks for clinical for chemicals, and they are funding for validation of the methods. They are putting about $400 million on a 7-year project. And the Canadian Environmental Protection Act 2023 also prioritize the new approach methodologies for chemical and drug safety testing. So with this, I will go to show you some of the techniques, how we make the tissues. Next slide, please. So how do we make these tissues? For airway tissues, for chemical toxicity, that's the topic of today's talk. So we just take lung samples from humans, and then we isolate the cells, expand them in culture, and then we cryopreserve them for long-term storage. And from there, we take vials and we sit them onto a cell, onto tissue culture inserts, and using the specialized media, we make them differentiate and certified to make a fully differentiated tissue structure again. So that is the whole technology that we're developing, we have been using, and to produce reproducible and reliable tissue models. Next slide, please. So the human airway tissue models that we develop over here at MatTek is an in vitro 3D organotypic model of that's obtained from the human tracheal bronchial epithelial cells. The cells are grown at air-liquid interface. What it means is all the feeding is from below, and the upper portion of the tissue is exposed to air. And we have more than 30 donors of airway tissue models, and they're with different backgrounds, like healthy, COPD patients, asthmatic, and smokers. And the tissue models that we produce are highly reproducible, stable, and they can be shipped out throughout Europe and Asia. So some of the techniques, the cells that we produce, as you can see, they have ciliated cilia formation on the surface, and they have mucociliary epithelium, and they are grown on a microporous membrane. Next slide, please. But for translational purpose, the humans can be compared with the humans. but most of the data are collected in rats. So we have to bridge that gap of translational biology. So what we are trying to do is we are developing rat models for cross-species comparisons, and we use this as a translational tool to study chemical screening, drug permission, and this research. As you can see, the failure in animals to predict human responses underscores the need for new methods for toxicological research. Next slide, please. So this a very nice picture. So it's a false color, but it's a scanning electron microscopy of the airway tissue models that we developed here at MatTek. And this shows the villi formation by electron microscopy. So this is for clearance and things like that. So it has a lot of functions. So we are modeling the right way as we see it in the human body. Next slide, please. So the problem for inhalation is that inhalation toxicity accounts for about 15% of occupational diseases globally. So MatTek airway tissues have been has been used for physiological exposures to pathogens, chemicals, or therapeutics. It's ideal for this type of applications. And the airway human relevant biological response makes it ideal for respiratory disease, chemical risk assessment, and drug development research. And with that, we will go to the inhalation toxicity part that we are going to discuss with this new project. Next slide, please. So we have animal models. We have also in vitro human models, so they can be compared. So the animal compared to animals, the human compared to animals, and the in vitro models can compare to each other. So by making this mix together, now we can approach. We can predict human responses. We think that we can predict human responses in a better way than has been practiced for decades. Next slide, please. So this is how it looks like. So we have now 3D models. That's in the lower side, and the 3D in vitro animal versus human comparison can be made, and the 3D in vitro models can be used, incorporated with other technologies like in silico or computational modeling, as Mary mentioned it before, and that will enhance or accelerate the prediction, developing a prediction model to predict correctly human responses. And that will also allow us to compare animals to animals, and then to compare species extrapolation, whether that was the right way to do, or there could be other ways to do it. So these are the translational biology. We are now, because of the two different models that we are developing, we can approach this question very efficiently. Next slide, please. So with this tissue in mind, then we had a joint project with Charles River, and Charles River, with their expertise and their experience as the CRO, and MatTek, experience in tissue engineering. We combine our forces together to address one fundamental issue in inhalation toxicology. And for that, the first thing that we did was we harmonized the protocol for both labs, and the same batch of chemicals were used in both laboratories. And we have also even installed commercial instruments that has exactly the same instruments both in Ashland and at MatTek and at the Charles River Laboratories. And then we used four technical replicates and 6 dose levels to test the first round. And then we have controls. Then we have positive controls, untreated controls, and vehicle controls. And then we have used the common endpoints to predict toxicity of chemicals. Viability is one, as measured by LMR blue, and cytotoxicity of tissue, cytotoxicity of chemicals on these tissues by using LDH assays, and also we have been using membrane integrity measurement here as one of the endpoints. And also we collected histology, and we have isolated RNA for gene analysis and collected culture supernatant for cytokine analysis. And at least where the team meets weekly and discuss the progress of the project. In terms of cells, we have normalized the cells. So we used, for this test, we use normal human bronchial epithelial cells, 2D, and then the airway and human rat airway tissues. And we have acceptance criteria for the tissue to be used. They have to pass our quality control criteria to be used. And then we matched donor and passage number between the 2D and the 3D tissues. So then we start the tissues at the study at CRL and MatTek exactly on the same day. Next slide, please. So these are the 10 chemicals that we have been selected to use, and these are for selected based on the global harmonization system classification for identification and labeling of chemicals. That is a GHS classification for GHS acute classification. So you have seen some of the pictorial markers that you show it's dangerous and danger or warning signals on every chemical. So we have different classes of GHS, GHS classified chemicals as category 1, 2, 3, 4, and 5. So we try to see if we can predict these chemicals correctly using this in vitro assay method. Next slide, please. So the assay method is simple that we designed. So we selected 10 chemicals, and the first thing that we did was on 2D models on monolayer cells, and we did dose range finding experiments. We used 6 concentrations. We used 4 replicates per concentration, and we used a very matched protocol at Charles River Laboratory and MatTek. And for that, what we did was we take the cells, put them onto 96 well plates, and then we culture them 48 hours on both laboratories. And then after 48 hours, we wash the cells and we expose the cells with chemicals. And we once after 4 hours, we wash the cells, and we also collect culture supernatant for toxicity analysis and inflammatory responses. And then we allow the cells to recover for 20 hours. And from there, we also collect culture supernatants for LDH assays and inflammatory responses. And on the tissues themselves, we measure viability of the tissue after exposure of 24 hours. And then we also measure barrier integrity by tear. And also we also isolated RNA for gene expression analysis. So it's a very comprehensive test that we are doing. Next slide, please. So for the 3D tissue models, we also, we ship the tissues from Ashland to Charles River Laboratories. And after the packaging was unpackaged exactly the same day when the tissues arrived at Charles River Laboratories. When we get information that they are there, then we unpackage from the Ashland facility at MatTek. And on both laboratories, the tissues are cultured for 5 days for recovery. And then before we start any work on Monday, we start measuring the barrier integrity on both labs. If they pass our QC criteria, then they will be allowed, the cells are cultured for 1 day for recovery, and then we expose the tissues topically for 4 hours with these chemicals. And after 4 hours, we collect culture supernats exactly the same way as we did for 2D, and then we allow them for 20-hour recovery, and from there we measure viability, barrier integrity, histology, and gene expression analysis. And with that, I will take you to Mary to discuss the results that we got.
Thank you, Sion. So yes, I'm going to talk about our first objective. Next slide, please. So just to kind of show you how we're presenting data, I'm going to highlight just two of the chemicals we've used in the panel. So I'm starting off with the 2D exposures. So we used methyl isothiazolinone, MIT for short, because it's a lot easier to say, and propylene glycol, PG. So MIT is a synthetic biocide, and it's a preservative, and it's found in cosmetics, household cleaners. It's a hazard assessment, GH2, so potentially dangerous. And there are in vivo data for LD50s and no effect levels in rats and dogs. So that means we'll be able to, from our rat data, when we do it in 3D culture, we'll be able to understand that relationship between in vitro data and in vivo data. PG is, in contrast, is a non-toxic compound, and it's a common ingredient in foods, cosmetics, drugs, and vapes. And again, there are in vivo data on no effect levels for PG. Next, please. Next, thank you. So when we first get the data, this is how it's plotted as an example. So we're looking at single dose acute studies, 2D cultures, and results for MIT and PG for Alamar Blue. And it's a mean and standard deviation. So Alamar Blue detects mitochondrial activity. And if we look, the two graphs are laid out in the same way. So on the very left hand side is essentially the incubator control, and that's 100% viable. MatTek are in green, Charles River in blue. And the next column, formaldehyde, that's a positive control for toxicity. And of course, the cells are completely destroyed, so the viability is down at 0. Then as you go along the graph from towards the right-hand side, we're actually decreasing in dose. And it just reflects the way the study was laid out on the plate, in fact. So from high dose to low dose. So you can see that a couple of things to point out, that the data from the two labs are quite similar, which is always good, in the same ballpark, and that there's good viability at the low doses, and that decreases at the dose as the dose or the concentration increases. So we've got a dose response in both cases, and that if you look at the actual amounts, where we see a change from, let's say, 80% to 100% viability to less than that, then you're starting to see the toxic effect. And that is the point we're modeling. And in this particular case, we put the data through EPA software to predict what's called a benchmark dose, which is that dose where toxicity begins. Some people call it the point of departure. And as you would imagine, that the MIT dose is much lower than that for PG. So that's the first sort of analysis we do, and as we'd expect. Next slide, please. So the important thing to do is to look at the data as a whole. It's plotted as a graph. And I have to thank a statistician in my team for working in R and plotting these graphs for us and performing analysis. I've highlighted the MIT and PG here just to show you what the data look like as a whole. And that pointed departure movement from viability down to less than 0 viability, if you like. The data between the two labs are very concordant, which is great to see. They're pretty similar in the polyethylene glycol, PG as well, but maybe separation at the high dose, very high dose levels, which might be expected. And then if we're looking at the other compounds, again, there's good concordance, which is great to see. There's only one I pointed out in red, which is the isophorone diisocyanate. And clearly the concentration, which is causing a 50% loss in viability, will be different in these two, in this case. And this was very useful for us to see, because it drove extra discussions and more review of how the actual chemical was made and when it was administered in relation to when it was prepared. And in fact, the protocol was tightened up, and that problem was solved for the 3D cultures. So again, although we're coming at this method development and standardization, both companies, CROs, and this bread and butter of what we do, there are still things we will learn along the way about standardization that we will be able to bring into the protocols and eventually publish. Next slide, please. So once we've done that, the data, in this particular case, we went for an EC25. That's an effective concentration 25. So that's the early part, just after, if you've got stable, viable, and when you've kind of got a 25% effect, difference from the baseline, if you like, and the data presented as mg per mil. And the first thing, so the compounds, the chemical list is now being listed in the GHS acute grade, going from very toxic to non-toxic. And the first thing that we obviously wanted to see was that the values between CRL, the EC25 values between CRL and MatTek were similar. And generally that's the case. So all very similar. So that was, if we look at potassium dichromate, they're the same. Methyl isothiazoline, about the same, etc. So as I pointed out in the graph before, the isophorone was different between the two labs, but we located that error down to the formulation. And the other thing that surprised us, because the other observation to make is that the mg per mil for the EC25 increases were decreasing toxicity, which is as expect. That's not true for the butyl 1,4-diol. And this is a compound that's used in manufacturing and it's at low concentrations in cleaning fluids. So that's out of sync with the other concentrations in relation to the GHC grading. It was repeatable across labs, and we actually repeated that, and that is a very reproducible result. And at the moment, we don't actually have a reason for it, but hopefully in the fullness of time, we will sort that and understand why, to inform how you'd use these assays for unknown chemicals. Next, please. So now that's the 2D data. So that's the 2D data summarized. So now we're moving on to the 3D data. And this slide's a busy slide, but taken example data from lactose. It's the mean and standard deviation. It's non-toxic. On the very left-hand side is the 2D LMR viability assessment. And on the right-hand side are the graphs of the human tier, because now we have tight junctions, we can do tier and we can do LMR blue in the 3D cultures. And generally speaking, if we're looking at the 3D culture, human and rat, that there is no specific dose response at the concentrations we use. And that was going from 450 mg per mil down to 1.85 mg per mil. And it was pretty much adding on as much as we could add of lactose. And that's a good result. And if we compare between labs, between rats and mouse, rat and human, they're about the same. So that's good. And just for comparison, the 2D were more sensitive to lactose at the very high dose. So we were able to get an EC25 and EC50 for the 2D. Again, in the literature and what others have found and what we've found in the past, that 2D cell cultures are more sensitive than 3D cultures. And that would certainly be anticipated based on the multicellular nature and the more, let's say, in vivo-like cellular arrangement of the 3D model. And we move to the next slide, please. So now the same sort of approach, but this time we're looking at MIT, the mean and standard deviation, and it's a toxic compound. We have the 2D just to remind you where the dose concentrations are and where the changes occur, reduction of viability occurs. And again, we see the reduction in viability in the 3D human, whether it's tear or Alamar blue assay, so viability assay. And so when we then kind of work out the EC25s for, and I've just highlighted maybe the key numbers here in the table. So we have the human 3D Alamar blue. For Charles River it was 0.55 EC25, and for MatTek it was 0.773. So again, quite similar. And in the rat, they're in the same ballpark, but tended to be higher, both for Charles River and MatTek. So we have confirmed that in this case, that the 3D model is less sensitive than the 2D model, that we can get reproducibility between the two labs, and that the rat model is not so dissimilar from the human 3D culture. Next slide, please. Again, we've combined all the table, the EC25s from the human model, both CRL and MatTek, into one table. Again, the chemicals arranged in increasing order of toxicity. And as we've seen in the 2D, generally, most of the chemicals, there's reproducibility between the two labs, which is great to see. There are few sort of corners of the issues we'll have to iron out. And I'll just point that out, that silica in the MatTek lab, for some reason we found toxicity with this compound. We expect that will be a formulation issue, because the tear, it possibly the formulation may be interfering with the assay rather than the cells, because the tear between the two labs was quite similar. So that's something for us to work out for future. Again, the isoflurone was a higher EC25 than expected based on other compounds that maybe the hazard level 2. We don't have an answer for that yet. We'll need to see how the data model, maybe when we understand the rat in vitro to the rat in vivo. Well, so that might give us an indication of understanding that relationship between hazard assessment. It may indicate there are other elements affecting the toxicity or the action of this chemical in the mechanism of toxicity for this chemical may be more than maybe directly damaging aspects or necrotic or damaging the membrane of the airway epithelium. But that is to be something to be considered. Next slide, please. Thank you. So our acute experimental data summaries. So to summarize, the 2D human monolayer cultures were more sensitive than 3D for most chemicals, and the range, it was difficult to predict the range of this sensitivity. We may be able to infer that if the chemical is toxic, there may be a narrower range ratio for 2D to 3D than if the chemical is non-toxic. I mentioned the exception of 2-butyl 1,4-diol. We got reproducibility between labs, and generally 3D human EC25s generally ranked in the order of the GHS, acute respiratory hazard, and that the EC25s for the rat model was slightly higher than the human tissue. And based on this preliminary assessment, we've provided EC25 concentrations that map against a hazard class prediction. I think in the fullness of time, when we have our complete data set, that we may revise what we may qualify or develop this table further. The final slide before the questions. So differences between labs can be linked in part to dose preparations in some occasions, and that these procedural differences were identified and harmonized for the 14-day study, which is just completed. We found that the dose setting in the 2D model was generally useful for the 3D models. So that is rather than going in completely blind to the 3D model, a 2D and a cheaper model can be very useful in that regard. That we are hoping that the rat model will be a tool to aid translation between in vivo rodent data and in vitro human outcomes, and for model predictions. I've talked about the E2E25 today for cytotoxic endpoints, but we're going to be overlaying and qualifying and a better understanding toxicity by also looking at cytokine release and gene expression, and also more, looking at the morphology of the 3D culture, and we will be modeling eventually the human exposure using these computational fluid dynamic models and multiple particle dosimetry models. The final picture I've pulled in here is from the discovery pathology team, where they've used a Visiopharm algorithm to start quantifying the total cell area, the total, which is in green, the cilia, and also the intracellular spaces that can occur in this particular model. And we expect aspects like the thickness to change with toxicity and the extent of intracellular spaces to change. Thank you for your time, and I think we can move on to the questions. And very importantly, thank you everyone who's, as I said, this is a very collaborative grant and approach. So want to thank my team and Joe in particular, the inhalation team, the discovery team at Charles River, and also the MatTek team, particularly Kalyani, who's been working hard at the coalface. So thank you.
Hey, thank you so much to Dr. McElroy and Dr. Ayuni for a great presentation with lots of excellent data and wonderful insight into the testing landscape currently and the future of it. So we'll move into a brief Q&A session. So I have one question here. Is it normal to observe little differences in toxicity between rat 3D in vitro culture versus human 3D in vitro cultures using the same compound testing condition, considering rat versus human in vivo probably have different tox ranges?
Well, I think that's a great question. I think the number of times that I think we'll explore all those different assumptions, I think, in that, because I think the number of I think the times, and Sayum will know to a greater extent than me, there is good concordance between the two systems. There will be differences, but from what we know so far, maybe the extent of toxicity is similar in this case. We are just looking at this extent of toxicity. When you move into in vivo, there will be other aspects to consider, possibly immune aspects. So what's normal yet has to be defined, I would say.
Yeah, I think some of the things that probably toxicity there may be similarity in toxicity responses, which we have published that the previous publications that we had showed that there was about 88% correlation there, but in terms of we are doing a whole set of assays like cytokine analysis, gene expression analysis, looking for pathways and things like that. Say, for example, if those are where we are going to see more differences in terms of between the rats and the human in vitro tissue models. So in general, for inflammatory responses, the rat models were not that great. So everything that passed for inflammatory responses failed in human clinical trials. So there are differences there will be different. We assume there will be differences in cytokine responses and chemokine responses, and also gene expression levels.
Yeah, And just a point, this is just acute exposure. So
Yeah,
when we come to do the repeat dosing,
we make a difference too.
Yeah, exactly. Yeah.
Thank you both for that answer. It looks like Joe Wallace is in the comments giving us an assist with answers, so thank you very much, Joe. And we had a question, what rat strain was used in vitro? And Joe provided a great response that no in vivo testing was conducted. The rodent testing was not conducted. It came from pre-published data and online sources. So there were no live animals used in these experiments. I just want to clarify that to viewers.
Oh, yeah.
And this person also asked what strain of rat were used in the generation of rat epi-airway. I don't know if you can answer that, Sayum.
The Sprague-Dowley strains were used. That's what my record shows.
Okay. And then there are a lot of expertise in the comments in the questions, but one question is asking if certain results suggest the difference in 2D and 3D responses are a result of a metabolic difference. I don't know if you can access the chat to read some of these questions. That one might be.
Yes, there's lots of differences between 2D and 3D. One is just a monolayer, and the cells are bathed, if you like, in the apical surface is bathed in the solution with the test article, whereas in the 3D model we're putting a volume, in this case 50 microliters on top, and it takes time for the compound to permeate through as it would do in vivo. I would say because the compound, the 4-butyl 1,2-diol, I mean, it is a toxic compound, and that was detected as more toxic in the 3D than the 2D, then obviously we've got better representation in the 3D, just from this acute application. There may well be other differences, but it may at this point be structural and accessibility, exposure, which will also affect the ability to metabolize too downstream, but it's about exposure and how the chemical gets to the cell.
Thank you for that answer. And another attendee asked to recap, we have a set of 10 compounds that may serve as reference compounds for this test. What additional compounds are you considering adding?
Yeah, great question. I think we'll have to think about that, because I think when you come to look at the toxicity of the chemicals and the mechanism, there's not always a great deal of information. So I think in part of what we're doing, we'll also build up a profile of toxicity for category 1, 2, 3, and 4 in terms of the cytotoxicity, maybe their inflammation profile, is the gene expression, as Theo mentioned, and the histological effect. And it would be quite useful to have kind of a multi-parametric approach to looking at toxicity. We're looking at the membrane, looking at the cell function, the ability of the membrane to be an effective barrier, but we also will now be looking at what's happening maybe before toxicity is manifest in this damage to the barrier. And there will be differences between the category of chemicals. So I think what we do and how we describe this toxicity and maybe even the pre-toxic effects will help build up a picture, and that might inform what we might use as reference items in a wider screen.
I think we can use. We are looking for chemicals from EPA list of chemicals.
Yeah.
So that way we can if we are going to take it directly for validation purpose with regulatory bodies, that we have to work with them. So they have a list of chemicals that we can select, and that's where we are going to base our assays. We have in the phase 2, we are going to test unknown chemicals so that we can test our prediction models that were coming up. So for that purpose, we will have some chemicals from EPA list that we can choose.
Great, thank you so much. Another good question, is there, I think, Sam, you can provide an answer to this. Is there eventually going to be an assay developed to investigate uptake across the alveolar cell in the 3D model to bypass the use of animals and determine absorption into the bloodstream via lung tissue?
Yes, that's next, but it's one by one. First, we have to we have to convince the regulatory bodies with one organ, and then see this organ can. Once we get acceptance like that, and then the test methods are get acceptance, then we can it is open. We can do alveolar, we can do other we can combine all the three nasal, bronchial, and alveolar tissues at the same time and test it through the respiratory tract. So all those are possible, but we have to start from one, and that is what we choose.
Yes,
the airway.
Yes, I'll make a comment there. So I'm not from a tissue developer perspective, but for someone who's worked with the alveolar epithelium previously, yes, it is definitely, it's the major surface lining cell of the respiratory of the lung. So it's a really key cell to model. It's also extremely different. It's a very large cell, has multi-cytoplasmic blades that extend across different alveoli. So it's if you that old idea, that the important idea that structure reflects function, it's the next level of model development, I would say. And in order, the upper airway, it's quite a good model for the human upper airway in vivo. So there's back to that ICVAM framework, the biological definition of what you're doing, if you can justify it, is a very strong first step. So I think there will be greater challenges for the alveolar. They are being made, but yes, that's kind of another step. Technological advancement is required.
Absolutely. Thank you both for that. A good question. Are you thinking of developing these models with other species used for preclinical testing? And do you see the rat model becoming commercially available? The rat model is commercially available.
Yeah, it is. Yeah, now we are expanding that model to the animal models to include primates. So we had a contractual agreement with NIH and cats, and we have developed the airway tissue model from rhesus monkeys, and so that is now we have developed that, and also another pharmaceutical company was interested in developing airway tissue models from African green monkeys, and we had a contract with them, and we have developed that model for that particular company also. So now the different animal models, in vitro animal models, are available with us.
Thank you very much. I'm just picking through some more questions here. Joe Wallace, thank you so much for jumping in to help with the answers. We're so tight on time. We're already a little bit over, but I have a couple of more broad questions for both of you, kind of challenging your expertise and your experience in the field. Mary, this is probably a question for you. Charles River has been a big stakeholder in animal testing. Why the shift towards adopting alternative methods?
I think it's seen. Well, I know it's seen as inevitable, but I mean, I've seen the phrase that we're not, and I think it reflects really where we are, that we're phasing in alternatives, human-based alternatives for human-based risk assessment, rather than phasing out animals at the moment. And I'd like, so I think that's the attitude, that these in vitro models and the development of AI can add so much mechanistic data to tox assessment. And in fact, I was privileged to be part of a BioSafe committee that just review on the use of NAMs, and the paper's just been published in Drug Discovery Today on the use of NAMs that replace large animals for the development of those therapies that are so human specific that the tox species are just completely not relevant. So in this case, the developers, that's the pharma companies and the regulators, on a case-by-case basis, came up with essentially a NAM strategy to decide on a safe dose for first in human dosing. So it is happening, and I've used the term NAM creep. I know, as in ivy, that these models are being used when scientifically justified. And in this particular case, it was for serious and life-threatening diseases. So there's definitely an important commercial drive to make use of these models and make them acceptable for regulatory review and decision making. So thank you for that question.
Thank you. And I think we'll just do one more question that I think, again, taps into your expertise and your experience in the field. And either one of you or both can provide an answer. Do you think that there will be greater regulatory acceptance of NAMs models in the future? And what does that timeline look like? It's a tough question.
Yeah, I can say something, and then Mary can come in. So yeah, there is a great push, particularly with NIH. NCATS has a great initiative that both the regulatory bodies like NCATS, FDA, and some other organizations, they are putting their effort together to come up with NAMS approach to evaluate NAMS approach, and they have initiated a 5-year project. They are going, according to their plan, they are going to fund it for 5 years. So they are going to have five multi-center labs that they want to fund for 5 years to address this question. So the whole concept is that taking it from the in vitro work, evaluating the methods, and then take it working in close collaboration with regulatory bodies. That way, at the end of the project period, probably those are they will be accepted as some of them will be accepted for by the regulatory bodies. And also there are independent moves. For example, we are working now with working groups that we have formed working groups for validation studies for a couple of MatTek tissue models. That's ongoing process, and there is some kind of a movement in that area also. I mean, some interest, but probably in the coming 5 years, 5 to 7 years, probably we'll have those models accepted. But right now, for some of our models on a case-by-case basis, there is acceptance by the FDA, and hopefully that will expand because of this as a new act, as a new bill from the regulators. Probably that will push pressure and enhance the timeline for accepting these models. Mary?
Yeah, I'd say, well, there's no one has a crystal ball on this, but I mean, they are being used already, as I mentioned. I think in the lung space, and I should have mentioned in the talk, there's the chlorothanocil OECD case study, which is driven by Charles River, not Syngenta, and Charles River helped out with Clive Roper, and Joe was involved in that work. And that case study replaced a 90-day rat inhalation study. It's a case study, so it's not a regulatory guideline. And on the back of that, I think the EPA takes a case-by-case approach for suggesting the 3D lung organotypic cultures for risk assessment alongside the animal work. So it is happening, and it's kind of happening below the radar to some extent. But I think it will, in order to replace, let's say, a 90-day tox study, that's multiple organs, multiple methods and cell types of toxicity. we just don't understand enough the interrelationship of organs and the systemic exposure, and let's say even the metabolic size of organs and how they interact in order to model it in vitro yet, in my opinion. But the science is developing, it will happen. But yeah, timelines. So timelines, it's already happening. I think they're already being used in the discovery space to better inform candidate selection. So that means that the drugs that go through to animals will be already de-risked for human toxicity. So I think they're probably already making a positive impact on the whole safety assessment space. Thank you for that question.
Thank you very much to both of you. And I think it is certainly the hope of us presenters and a lot of people joining the call today that we will see more use of NAMs as time goes on. And of course, it is very difficult to put a timeline on that. We are well over time today. So if we did not get to your question, we will try to respond offline privately and give you answers. Everyone will receive a recording of this along with our SOT poster on the first phase of this project that was presented at the meeting last month. And you can always reach out to us if you have any other questions about this project or about MatTek and Charles River, what we offer as services and products. And I hope everyone has a wonderful day. Thank you again for joining us this morning. It's been a pleasure having you all. Thank you.
Thank you.
Thank you so much.
Thank you.